Andy, can you hear me all right? Good, all right. Just don't fall asleep, okay? Amanda's in bed. She had a blanket over her with a computer on her. No, I definitely heard that. Amanda, I sent the quiz to you via email, and I also sent a handout via email that I'll be referring to today. Sent it, 10 minutes ago. You should have received it by now. Hey, Dave.
Hi, how are you?
I'm trying to stay warm. I sent that quiz to you.
Yeah, I got it.
And also a handout that I'll be referring to today.
Yeah, I got him.
Okay, good, good. Amanda's joining on Zoom. She had her first round of Pfizer, and she's a tad under the weather. So
Yeah.
I'm pleased this punch she was able to get it. So
Oh yeah, no, that's real good.
it's a non-reactive reaction. So 2 carbons, put them together, 2 carbons, put them together, that part. Okay. Let us go ahead and begin. Very good. So we discussed the synthesis of carbohydrates. Now we'll talk about how you make fatty acids. And then from there, how those fatty acids are assembled to form lipids. We'll look first at the phospholipids, glycerol phospholipids. Then we'll look at sphingolipids. Then from there, we'll look at lipids that are used in signaling, namely eicosanoids. So to make a fatty acid, you're going to begin with this molecule, malonyl coenzyme A. And if this looks like acetyl coenzyme A, that's because it should. Notice the only thing malonyl coenzyme A has that acetyl coenzyme A doesn't have is this terminal carboxyl group. And to put that on, so let me use my pen here. So this right here, that's the only piece that's different. So this is made from acetyl coenzyme A. And the synthesis of malonyl coenzyme A is essential for the synthesis of fatty acids. Consequently, this step where you make malonyl coenzyme A is very heavily regulated. Oops, wrong way. So Malonyl coenzyme A is made from acetyl coenzyme A by acetyl coenzyme A carboxylase. Acetyl coenzyme A carboxylase, which is given the abbreviation ACC usually. So this has 3 different functional subunits. Because it's a carboxylase, you might guess that it uses a specific cofactor, that being biotin. And you would be correct if you thought that. So we've seen this before, the A, B, C structure of these carboxylases. This one is very similar in that regard. So you have the biotin carboxylase. It has the lysine that, excuse me, the lysine is actually found in the BCP, that's the B subunit, and then you have the trans carboxylase. So this is called A, it's called A because it's the acceptor. And then B, this is the biotin carrying subunit. And then this is C. Why is this C? Because that's a carboxylase. So once again, you need to use ATP. ATP is going to activate the CO2, so that the CO2 can attach to the biotin. This then is going to swing between here and here, picks up the CO2 over here, swings it over here, and then it will carboxylate its substrate, acetyl coenzyme A. And the product is malonyl coenzyme A. Malonyl coenzyme A. All forms of acetyl coenzyme A carboxylase contain the biotin prosthetic group, all of them. In bacteria, this enzyme is a complex of 3 different polypeptides. In plants, well, rather in animals, it's a single multi-functional polypeptide with 3 different domains, the A, the B, the C domain. Plants have both kinds of enzymes. The enzyme that is bacterial tends to be associated with chloroplasts. The enzyme that is multifunctional tends to be in the cytosol of the plant cells. And as we said, all forms contain that prosthetic group. All right, so you've made your malonyl coenzyme A, and now you're going to use that to make fatty acids. And the synthesis of fatty acids kind of looks like the oxidation of fatty acids in reverse. And if we look at this picture from our book, well, these KP95s are good masks, but holy Moses, they're warm. It's so hot under here. It's just, ugh. All right, so if you see here, the enzyme that makes fatty acids is a complex called fatty acid synthase. Fatty acid synthase. It's going to have 2 sites. One will be the acceptor for the acetyl group here. The other is going to be the acceptor for the malonyl group. You'll have a condensation. So really, this group here does not get incorporated in the fatty acid. That group exists largely to activate the acetyl unit. So you're still going to build the fatty acid 2 carbons at a time. Remember, you broke down fatty acids 2 carbons at a time. Now you're going to build them 2 carbons at a time. So this is going to be lost here. So you get condensation. Here's the carbonyl carbon, remember, slightly positively charged. Here's this methylene carbon, ever so slightly negatively charged. You get condensation, and the CO2 will be displaced again. CO2, very good leaving group. Now you have the beta keto group. You'll reduce it. So whereas before you had a thiolysis, then you had, before that, you had an oxidation. Now that oxidation is reversed, it's a reduction. Notice for biosynthesis, we're going to use NADPH. That produces the beta hydroxy acyl. Then we're going to take a water out. We have a dehydration. That produces the NOEL. And then we're going to reduce that again, using once again NADPH. And now we have the proper saturated acyl group that's been elongated by 2 carbons. Wash, rinse, try again. And as you can see, this back and forth, back and forth, back and forth. You keep adding malonyl coenzymase. You grow this by 2 carbons, add another 2 carbons. Pretty soon, you're going to have a carbon that is long enough to be released. And that's how you're going to do this. So it's a cycle. You run the cycle again and again and again. Every time you're in that cycle, your fatty acid grows by 2 carbons. Yeah, goes back to the beginning, fast as go, collects $200, and goes around the board again. Now here are the enzymes involved. There's 7 different polypeptides, 3 different accessory proteins, and they are mercifully abbreviated. So you have the acyl carrier protein, ACP, acetyl coenzyme A, ACP transacetylase, which is abbreviated AT. Beta keto ACP synthase, KS. Malonyl coenzyme A, ACP transferase, MT. Beta keto acyl ACP reductase, KR. Beta hydroxy ACP dehydratase, HD. And NOEL ACP reductase, or ER. And these, again, in E. coli are separate polypeptides, just like you see there up at the top. 7 different activities in 7 different polypeptides. Notice at the center of that pinwheel is ACP. This is the fatty acid synthase complex. Fatty acid synthase complex.
Should we know the proteins by their full names or
Yep.
by their abbreviations, or both?
Both. Both. Now, in yeast, many of these activities are encoded by one polypeptide. So you could see the NOEO reductase and the beta hydroxy hydratase, sorry, the beta hydroxy, beta hydroxy, the hydrase, ACP transferase. These are all one polypeptide. Same thing with the synthase, the beta keto ACP synthase, and the beta keto acyl reductase. Those are one polypeptide. And then the malonyl ACP transferase, that's one peptide. And ACP is one peptide too. So there's a trend here. You're starting to fuse these into single polypeptides. In vertebrates, it's one big, huge polypeptide with 7 different activities. Because these all do the same thing. Now you have one gene to regulate, not 7. So there's a bit of economy about it, if you will. Two separate polypeptides, 7 different activities. Because this malonyl ACP transferase is part of this polypeptide here that does these as well. Okay. So those 4 different polypeptides, 4 different activities that map to one polypeptide, and then the other 2 in the yeast, you've got 2 genes essentially that are co-regulated. But in vertebrates, you just have the one. So your building blocks, as we said, consist of malonyl coenzyme A. It is transferred to the acyl carrier protein, or ACP. ACP contains a 4-phosphopantothene, 4-phosphopantothene prosthetic group, and your fatty acyl chain will be attached to the ACP by virtue of a thioester bond. That means that your transfer from malonyl coenzyme A to the ACP doesn't require the input of energy because both bonds have an equivalent amount of energy. So here's the ACP polypeptide that is designated by this big sphere. Here's your pantothene, and the pantothene ends in a sulfhydryl bond, sulfhydryl group. So that's going to form the thioester with the malonyl group and so on and so forth, or the acyl group or whatever. This pantothenic acid arm, very highly flexible. That's why it's able to pass essentially its activity to this one or to this one or to this one or to this one. You get the point. It's because of that flexibility you can have this arrangement of those enzymes. All right, so we'll pass the substrate from one enzyme in the fatty acid synthase complex to another. So you start by charging up the two thiol groups in the fatty acid synthase complex. That includes ACP and the beta keto ACP synthase activity. Both of those contain thiol groups. So ACP and then the KS. Those two thiol groups there. To the KS group, the beta keto ACP synthase. The beta keto ACP synthase is going to get an acetyl coenzyme A, and then the ACP is going to get a malonyl coenzyme A. Acetyl, that's to start, that's to begin. So you need an acetyl group, acetyl coenzyme A group. You need that to start. Once you start, you're never going to use this again. You're just going to use malonyl coenzyme A. So the malonyl coenzyme A, ACP transferase, so-called MT, will attach the malonyl coenzyme A, the malonate from it, to the thiol group of the ACP. Complex is charged. It's ready now to go ahead. And here is the sequence of reactions. We'll go ahead and break these down into one at a time. I just want to show you where we're going. All right. Okay. There. Okay. All right, let's walk through this. So you charge these two up. Your malonyl coenzyme A ACP transferase. It's the malonyl coenzyme A here. The beta keto synthase, beta keto acetyl coenzyme A synthase, charges that. You're ready to go. First step is a condensation. You're going to condense this group with this group, and you'll lose the CO2. That's your first step. All right, and your this KS group is going to be responsible for that condensation, the beta keto acyl ACP synthase. Then the next step, you now have the beta keto acyl. Now you're going to reduce that. You're going to reduce that. NADPH will be involved. You reduce the beta keto group, you turn it into the alcohol, and the beta keto ACP reductase is responsible for that. You now have the beta hydroxy acyl, butyryl group in this case. Next, you're going to dehydrate the group. Take this and a hydrogen, take the water out, and you make the NOEL. You make the NOEL. So that dehydration is the result of the emergency room. Sorry. It's the beta hydroxy acyl ACP dehydratase, HD, No, that's the next one. HD. You know what might help? Let's, okay, I'm gonna take my, do the highlighter. All right, so the first step here, that's this enzyme right here. Next one is this enzyme right here. And this attachment here, that's the work of this enzyme right there. Now I get to go back to school and color again. And then this reaction here, that's a result of this enzyme right there. The beta hydroxy acyl ACP dehydratase. Now we're going to reduce the double bond. This is different than fatty acid degradation because remember, when you made the NOEL, you used flavine. This time you're using NADPH again. So this is the NOEL ACP reductase. And then you are going to transfer this to here because this ACP needs to be ready for another malonyl group, and that is going to be the acetyl coenzyme A ACP transacetylase. Now we're back to here, and the malonyl is going to be attached by the malonyl coenzyme A ACP transferase, which is this puppy here. Yeah. And then this repeats again. You have a butyryl group. Now you add another malonyl group here. That's the work of this enzyme. Then your beta keto acyl ACP synthase is going to do the next bit of work here. Now you have another beta keto. Now it's a beta keto hexanoate. And the process just repeats itself. Cycling over and over and over and over and over again.
So whatever enzyme is highlighted is what's responsible for the next
Right,
step that
that's
happens.
the particular enzyme, or if it's a vertebrate, the domain of the enzyme responsible for catalyzing that reaction,
Okay.
that step in the reaction process. Now, once you get a fatty acid that is a desired length, there is a thioesterase that cleaves the fatty acid from the ACP. All fatty acids made by the fatty acid synthase complex are saturated, all of them. Now, usually the product is C16, it's palmitate. But work done in several laboratories has shown that you get a percentage of other fatty acids. So some stearate, C18, is made, and some myristate is also made. And then occasionally you get longer or even slightly shorter fatty acids. Those are rare. C16, palmitate is C16. Stearate, C18, myristate, C14. And then undecanoate, which would be C12, that's made occasionally, but very rarely. And then duodecanoate, C20, is really rare. If you need longer fatty acids, there we're going to see their so-called elongation strategies for making longer fatty acids. They're all saturated. So if you look at coconuts, coconuts make a lot of shorter fatty acids. People have studied the fatty acid synthase in coconuts, and they haven't really gotten a lot of information on what causes the fatty acid synthase to stop. So there is a signal. It's clear certain fatty acid synthases prefer certain sized fatty acids. How it makes those, well, what's different about the enzyme that says we want this, not this, is not known. Yeah, MCP oil, I'm pretty sure is coconut oil. And the coconut oil makes very good biodiesel. Yeah. Yeah. It soaps, a lot of soap products will have coconut oil in them. You can smell it. In fact, when you go to the baseball field and there's that smell of popcorn, that smell is actually coconut oil. Coconut oil is used to pop popcorn at the ballpark. So fatty acid biosynthesis requires a lot of reductant and a lot of ATP, because it takes ATP to make malonyl coenzyme A. So if you want to make palmitate, you need 14 NADPH, 7 ATP, and 8 acetyl-coenzyme A. So this is a very expensive enterprise from an energetic standpoint, but it's essential. Absolutely essential. Phosphate. Phosphate. NADH, NADPH, the difference is a phosphate.
Right, but on this slide it has the plus hydrogen.
Yeah, because like NADH, it just carries 2 electrons, 1 hydrogen atom, hydrogen ion. So it carries it as a hydride. Same thing with NADPH, the other hydrogen atom goes into solution. Not like flavings that carry both. So the location of fatty acid synthesis differs between animal cells, yeast cells, and plant cells. So in animal and yeast cells, and frankly fungi too, fatty acid synthesis is a cytosolic adventure. And then the synthesis of your lipids for the membrane, sterols too, also both desaturation and elongation machinery that we'll talk about pretty soon are in the smooth ER. Yeah. In plant cells, fatty acid synthesis occurs in the stroma of the chloroplast. And fatty acid oxidation occurs in peroxisomes. You don't have fatty acid oxidation in plant mitochondria. You do, however, have fatty acid oxidation in animal mitochondria and in fungal mitochondria too. You also have some fatty acid elongation. You also have ketone body synthesis. You do have acetyl coenzyme A production in plants and the Krebs cycle and what have you. But fatty acid synthesis in plants occurs in chloroplast because that's the site where you have a high NADPH to NADP ratio, just like in the cytosol in animals. That's why you move this to chloroplast, because you make so much reductant via photosynthesis.
So it goes where
Yeah, precisely, precisely. So fatty acid synthesis and beta oxidation occur in different places in plants and animals. So again, fatty acid synthesis occurs in the cytosol in animal cells, in us, in yeast, filamentous fungi as well. In plants, it occurs in chloroplast. Chloroplast. The stroma, yeah. The soluble bits. Now, if you want to make fatty acids, you need acetyl coenzyme A. Where is acetyl coenzyme A made? It's made in the mitochondria. Does it cross the mitochondrial membrane? No. So you need some sort of strategy to shuttle it from the mitochondrion to the cytosol. And here's the means by which you shuttle it. You use citrate. You use citrate. So this is the mitochondrial matrix. This is the cytosol over here. So let's go through this. So citrate synthase, that's a Krebs cycle enzyme. No surprise here. Condensing acetyl coenzyme A with oxaloacetate, that's a Krebs cycle venture. No surprise there. Citrate then can pass through a specific citrate transporter into the cytosol. There, it will experience a cytosolic enzyme called citrate lyase. Citrate lyase. Citrate lyase undoes this reaction here. So you get acetyl coenzyme A, and you get oxaloacetate. Oxaloacetate will be reduced to malate. Malate. So there is a cytosolic lysozyme of malate dehydrogenase. So citrate is disjoined and in order for that reaction to occur, and then from that you get this is oxaloacetate. This is citrate lyase. What on earth is that? Now there's two ways to skin this cat right here, getting this back into the mitochondria. So the first is shown there with well, first you're going to convert it to malate. And you can see this also will use NADH just like the cytosolic enzyme. That's malate. All right, so malate can travel into the mitochondrion via the malate alpha ketoglutarate transporter. This is a transporter we've seen before, not an issue. Alternatively, you can use something called malic enzyme. Malic enzyme. This is a really important enzyme. So this can pass through. But you need reductant in the cytosol. You've got some here. You can also get some here. This enzyme is malic enzyme. Malic enzyme. Malic enzyme is going to convert it to pyruvate. So you convert this to pyruvate. And then this can go through the pyruvate transporter. Pyruvate, that's easy. Because pyruvate, you can simply use pyruvate carboxylase to convert it to oxaloacetate. Malate then can just go straight into the Krebs cycle like you'd expect. And then that just condenses with acetylcoenzyme A. And then that just makes citrate. And citrate can go through. I'm running out of colors. This transporter here, that's citrate synthase. Otherwise, malate can be converted back to oxaloacetate by malate dehydrogenase found in the cytosol. Slightly expensive. Well, you're leaving, so you're leaving reductant here. You're leaving reductant here. You're burning an ATP here. I forgot to put that in. Because you're making a thioester from this, so you need to burn that ATP to make the thioester. Yeah, it's expensive. Well, most of the time it probably does malic enzyme, because that way you leave more reductant in the cytosol. And since fatty acid synthesis is such a reductant dependent process, you're almost certainly going to use that more than you will malate. All right. You guys can't see the blackboard. I drew this out. No, you guys can't see that. Well, the drawing on the slides is a lot prettier than mine. So, okay, how is this regulated? You regulate it at the beginning. And the beginning is the synthesis of malonyl coenzyme A. Acetyl coenzyme A carboxylase is the major regulatory step for this process. All right, so this enzyme, ACC, acetyl coenzyme A carboxylase, as you can see, is inhibited by glucagon and epinephrine. They will trigger the phosphorylation of ACC. When ACC is phosphorylated, it's not very active. Likewise, large amounts of palmitoyl coenzyme A will feedback inhibit this enzyme as well. Now, it's not on here, but let me say insulin triggers the dephosphorylation of this enzyme and its activation. So S's are hard. Insulin will positively regulate this enzyme and it triggers dephosphorylation of it. Also, citrate is going to activate it as well, because when you get citrate in the cytosol, that tells you citrate is bleeding from the mitochondria to the cytosol. That activates it. And citrate lyase, which generates more ATP, that's triggered by insulin as well. Yes, yes. In plants, when you increase the pH and also increase magnesium ions, that activates acetyl coenzyme A carboxylase. And an increase in pH and magnesium ions occurs when the lights are on. Remember too, citrate is a potent inhibitor of phosphofructokinase 1. So that prevents carbon from going through glycolysis. Instead, it keeps it in the fatty acid synthesis process. Phosphorylation of ACC causes it to form this complex that you see here in this picture. It lines up in this macromolecular assemblage of enzymes that are not active. That's what phosphorylation does.
So I for Amanda, does adding or She has to do that.
Yeah,
But
But-
it's not-
but remember, we talked about, Amanda, we're talking about you. She smiled. Remember Amanda said if she leaves the port in on one side too long, she gets this fatty lump, lipodystrophy it's called. That's due to the fact that insulin is stimulating fat synthesis. That's why you have to alternate ports so you don't get these buildup of fat where you leave the port.
I guess it's like a regular non-diabetic. You have like a multiple somewhere, you would also get.
Well, yeah, if you eat lots of sugar, you're gonna have lots of insulin, you're gonna make more fat. So insulin is a weight gaining enzyme, but it helps you build muscle too. So Dane, for example, when Dane lifts in the gym and then he recovers, he should eat protein, but he's going to need to eat some sugar too to guarantee insulin release. That way the protein from the meal he ate goes to his muscles, because insulin will tell his muscles to make protein from the amino acids. That's for your benefit, Dane. Now they're both smiling, so that's good.
We're both awake.
Yes, they're both awake. Yes, accomplished my task. So if you get a buildup of ATP, get a buildup of acetyl coenzyme A, you're going to need flux of citrate from the mitochondrion, and the phosphorylation of acyl coenzyme A carboxylase slows it down. That's triggered by epinephrine, and it's also triggered by glucagon. And then in plants, you've got this pH, magnesium wave regulating, which guarantees that the enzyme is only on when the lights are on. It's not on in the dark. Yep. Now in bacteria do not store fat. Bacteria store glycogen, they store PHB, they store polyphosphate. They don't store fat. You don't get a fat bacteria or something like that. So if you make fatty acids, they're only used for membrane lipids. So the regulation is a little bit different. In bacteria, you have the palmitoyl ACP, and that gets used directly for membrane synthesis. You don't need to hydrolyze it from the ACP. What if you want to make longer fatty acids? Say you don't have C16s and you want something longer for various reasons. Then you go to the so-called elongation systems. And they are present in the smooth ER and in the mitochondria, particularly in the outer mitochondrial membrane. So these elongation systems, these have been pretty heavily studied. They also include a condensation, reduction, dehydration, reduction set of reactions. Yeah, so this is how you'll make stearate and then longer fatty acids from the products of fatty acid synthesis. Instead of ACP, you use the coenzyme A linked version of the acyl group as a substrate. And so here is the way this cow eats the cabbage. So here's your acyl coenzyme A. Let's say that's a palmitate. So it's a palmitoyl coenzyme A. Once again, your substrate is malonyl coenzyme A, no surprise there. So you elongate, you use the so-called elongase. You'll generate coenzyme A and CO2, this one here. And you get a 3-keto acyl coenzyme A. These elongases, there's several of them, and they've been studied in some detail. That's the names of the genes that encode them. So SIG30 is, it was found in yeast, and it's functionally equivalent to so-called ELO2 in humans. ELO2, the Electric Light Orchestra 2. And ELO1 is specific for fatty acids that are either C16 or C14. And then EOL2 and 3 specialize in longer chain fatty acids. And then there's this SSC1. SSC1 is the yeast version of the human ELO3 gene. Interestingly, this one right here, ELOVL4, mutations in this gene cause an inherited form of macular degeneration. Oh, goodness. Just say degen. Just leave it at that there. Goodness, that's hard.
I think you get a gold star.
And then this one here, LCE, this is for really short fatty acids, C12s. So basically, you have elongases for fatty acids of virtually any size. Then you go to the reductase, 3-ketoacyl and coenzyme A reductase, the CAR. CAR will handle just about any size fatty acid. So there's only one of these. Then you're going to take out the water. You have the dehydratase that you see there. And then you have the trans 2,3-ennoyl reductase, or TER, which will reduce the trans 2,3-ennoyl coenzyme A. And now you've got your fatty acyl coenzyme A that's 2 carbons long. That's your elongation system. Remember, this is in the smooth ER and the outer mitochondrial membrane. Yep. Yep. Yeah. Very good. All right. Now, if we want to make unsaturated fatty acids, we have to subject them to the fatty acyl desaturase. Desaturase. This is also in the smooth ER. And this desaturase uses a set of B-type cytochromes. And these B-type cytochromes are an example of a mixed function oxidase. So these are going to make use of molecular oxygen. You have your desaturase, sorry. There's your B-type cytochrome and then a B-type cytochrome reductase. So the B-type cytochrome, as you can see, will be the one that will handle the reducing equivalents here. Hands those to the reductase. The reductase is a flavine. Flavine then gives those electrons to NAD. Yeah. Exactly. And then you end up making a water out of this. That's essentially where a lot of your reducing equivalents go. Is that water? Plant cells also have a desaturase that gives its electrons to a ferredoxin. Mammals produce double bonds at the delta 9 position. Mammalian cells can't do anything more elaborate than that. Plants can synthesize delta 9, delta 12, and delta 9, 12, 15. Plants can do things that mammals can't when it comes to desaturation. So pretty much at the delta 9 and before that, but after that, mammalian cells just can't make those. That's why we need essential fatty acids. Okay, looks like we're out of time. Yeah, we're out of time. We'll pick this up on Monday. Hopefully finish this out and then look at the synthesis of different lipids. We'll need your handout for that one. Very good.
Is our exam next week? Is it on the 19th?
It's scheduled for next Friday, yeah.
Yeah, it's scheduled for next Friday. Yeah, does that day work for everybody?
I think so.
Yeah,
Okay.
then that's when we'll have it.
All right.
All right then.
I'll see you guys see you guys on Monday.
Cool. Very good. I will see you then.
Yep.
Ciao.
Bye.